Hello, everybody. My name is Karsten Rinas, and I'm a PhD student at the University of Waterloo. And I'm working with the monoclonal antibody trastuzumab, and I'm going to present a poster on the effect of this antibody on the splicing landscape of different breast cancer cell lines. In generally, alternative splicing is a known hallmark of breast cancer, and for the HER2 positive breast cancers, the focus has mainly been on isoforms of the HER2 gene, and less on the total splicing landscape. Similarly, for the antibody trastuzumab, which targets HER2 positive cancer cell lines and limits their growth, their research towards its impact on alternative splicing is rather limited. Two cell lines were treated with their monoclonal antibody trastuzumab, which are the HER2 positive cell lines BT474 and SKB3. They were treated with antibody on day 1 and 4, and on day 7 their RNA was extracted and the library prepared, and their samples were sequenced with the Illumina HiSeq system. After the sequencing, the sample analysis for detection of alternative splicing events was very straightforward. We used for the alignment the STAR program, and then quantified the transcripts or exons with RSEM, and performed a differential expression analysis in general for RNA binding proteins with HR on the one hand, and on the other hand, we used the tool RMATs for alternative splicing detection, and then further performed an overrepresentation analysis with cluster profile and created sashimi plots to see the alternative splicing pattern for the exons. The analysis of the differentially expressed RNA binding proteins with HR is shown in figure 1, which is on the top left side of the results section. In total, 287 genes were detected which have RNA binding functions, and around 100 genes of these were differentially expressed in the BT474 cell line and approximately 30 in the SKB3 cell line. In dark green labeled, 6 genes were significantly changed in the same direction for both cell lines, of which multiple ones are known to be involved in alternative splicing patterns, which further supports that trastuzumab impacts alternative splicing pattern. In figure 2 on the top right side, we see a few summary figures for the analysis of the alternative splicing patterns. In A, we can see the Venn diagram for the detected significant events for each cell line, and see that they have an overlap of 41 genes, with BT474 having in total much more significant alternative splicing events than SKB3. And then on the bottom of that figure, we see on the left the volcano plot for the alternative splicing events for the BT474 cell line, on the right side for the SKB3 cell line. Next, we looked at the alternative splicing pattern of known oncogenes, and we showed here in figure 3 2 examples for HRAS and GRB7. For HRAS, we detected that the treated cell lines have a reduced inclusion of the exon 4, and for GRB7, we detected an increased inclusion of the exon 13. Both alternative isoforms are related to changed proliferation of their cells. The results of the overrepresentation analysis are shown in figure 4, and they were performed for literature-derived signature gene sets from molecular signature database. We detected 11 gene sets which shows a significant overrepresentation of events in both cell lines, and some of them are related to breast cancer or cell differentiation, which seems to be impacted effect of these alternative splicing events. Overall, we saw that the treatment of the breast cancer cell lines with trastuzumab has a significant impact on the cell splicing patterns. We detected change expression of alternative splicing related RNA binding proteins. We detected changes in external inclusion of important oncogenes and an overrepresentation of splicing events in cancer-related gene sets. Next, we plan on receiving nanopore reads and to have a more reliable transcript coverage and potentially providing a more reliable quantification. In general, thanks for listening to my recordings, and I'm wishing you a great conference for the remaining days.